Today, my topic of discussion is safety. One interesting topic: safety aspects of probiotics. Safety aspects of probiotics. Now, we all know that probiotics had very good health beneficial effect. Probiotics are live microorganisms, generally lactic acid bacteria and yeast also, and they have very good health beneficial effects. And there are some types, but when we are using this probiotic, uh, uh, we we trying to using this probiotic, we trying to use this probiotic, we have to keep this thing in our mind, to that whether we are consuming this probiotic, whether they are sufficiently safe for human consumption. This is our main question and main query about that. So we have to be sanguine, we have to be confident about this, that these probiotics are absolutely safe for consumption. So actually probiotics, we are there are three types of probiotics are generally available. um, Types of probiotics. Number one is lactobacillus and bifidobacterium. Lactobacillus and bifidobacterium blends. The bacterium blends. Second one is the Saccharomyces boulardii. Saccharomyces boulardii, one beneficial yeast, is also very good probiotic. Beneficial yeast. And third one is the soil based blends. Soil based blends, generally bacillus species. So these are the main three types of probiotics generally available. We are generally consuming these probiotics, and they are proved to be very good uh, probiotics. And also the Food and Drug Administration of USA has given the lactobacillus and lactococcus the generally safe as, and also bifidobacterium, so generally safe as, generally recognized as safe, means GRAS status. FDA has given lactobacillus, lactococcus and bifidobacterium the GRAS status. GRAS status means generally recognized as safe. FDA. Now, the FDA has given this status, GRAS status, and we have to be also careful about that, that these probiotics are not detrimental to our health. We have to take care of that, and we have to assess the risk associated this probiotic. Generally, I have told that lactobacillus, lactococcus, and bifidobacterium has no issue, but sometimes this bacteria can be proved very fatal, very dangerous. So we have to be careful about that. And I have previously also told that that probiotics are not suitable for some category of people, like pregnant women, like pediatric patient, and gastrointestinal surgery patients, and the liver transplantation patient, and the immunocompromised patient, like diabetes and the cancer also. So we have to be careful when we are trying to giving this probiotic, applying this probiotic to this category of these people. We have to be very careful about that. First thing is that. And what are the actually probiotics are good, no doubt about that. We can consume the lactobacillus lactococcus bifidobacterium without any doubt. We can consume these probiotics, but some new novel probiotics uh, are discovered, have been discovered, and we have to be careful when we try to apply these probiotics. Actually, probiotics are generally used as a nutritional supplement or dietary supplement. So they does not require any approval from FDA USA before marketing. But we have to be careful about that. And till now, FDA does not approve any health claim for probiotics. So this is the fact. But we know that probiotics is overall probiotics consumption is good for our health, is beneficial for our health. So we should not stop it. We should take this probiotic daily basis, but with care, with some pre-assessment, with some pre-risk assessment. We should take these probiotics, and those companies are marketing this probiotic in the market. They are also, they should be also very careful about that. There is no problem with the lactobacillus and lactococcus, but other novel bacteria, when we are trying to apply other novel probiotics like enterococcus, bacillus species, then we have to be very careful about that. And generally, the prescription for the probiotic intake, daily intake is 5 to 10 billion daily for children, and 10 to 20 billion CFU, means colony forming unit, for adult. Now adult, and generally 10 billion per daily consumption of daily intake. So 10 billion CFU daily intake of probiotic bacteria will be beneficial for our health. But in some cases like viral infection and gastrointestinal disorders, we should take 10 to 20 billion CFU daily, daily. So this is the general prescription for the probiotics. And another thing is the we should not apply this probiotics to the some category of people I have told, but also you should not we should not use this probiotic for long time. This is the another factor. For a time gap we can use it, but for long time it has some toxic effect also. So what are the uh, risks associated with the probiotics? We are trying to understand these things generally. um, first one is the first one is the toxicity. Toxicity, it may be acute, acute or chronic. It may be acute or chronic. Second one is the metabolic products produced by probiotics. Metabolic activity like bile deconjugation, bile deconjugation, bioamine production, bioamine production, D-lactic acid production, D-lactic acid production, mucus layer, mucus degradation, mucus degradation, like that. And another third point is the very important antibiotic resistance. Antibiotic resistance. Now, first point is the toxicity. I have told that if you take probiotic in large amount, there is a prescribed amount for intake of probiotic daily for children and adult also. But if we exceed this limit, then cross this limit, then there can be some problem. Some problem means like in the toxicity, some is acute and some is chronic. Acute toxicity means the colonization of the probiotic in the intestine and the translocation of the probiotic in the different places of the intestine, and the mucus layer degradation, like that, and the platelet aggregation. Platelet aggregation can cause some disease like endocarditis. Endocarditis, it is a disease of the layer of the heart, inner lining of the endo carditis. It is called endocarditis. This disease happen due to the platelet aggregation by the probiotic bacteria, and this is caused the endocarditis disease. Endocarditis disease is a disease of the inner lining of the heart, mainly heart valve. So there will be some problem of that, and some problem like nausea, vomiting, diarrhea. It can be happened due to the gastrointestinal disorder, like that. It can be happened. And another thing is the, if this probiotic will produce so many cytokines, means inflammatory substances responses. We know that inflammation is the our body's normal normal reaction to the threat, and it is the part of our immune system. We know that, but excessive inflammation materials like cytokines should not be tolerated, because this can causes some autoimmune diseases like arthritis, like cardiovascular diseases like high blood pressure, heart diseases, gastrointestinal diseases like inflammatory bowel disease, and the Crohn's disease, and also asthma can be produced, can be caused by this excessive inflammation. So we have to be careful about that, that there should not be excessive inflammation in our body due to the probiotics. And so chronic thing is not desirable, accepted in this case. So we have to be careful about that. This chronic diseases like endocarditis like diseases. and autoimmune diseases. And metabolic activity means bile conjugation. Some lactobacillus and bifidobacterium have the ability to deconjugate the bile salts like taurocholic acid, dicolic acid. So they can deconjugate these bile acids present in our intestine, and they can produce the bile acids from there. And from this primary bile acid, some secondary bile acid can be produced, and this secondary bile acids can maybe a carcinogenic effect. So this should be careful about that. Although this deconjugation of bile acid will control the cholesterol level in our blood, it is a good thing. Despite of this fact, we have to be careful about that excessive deconjugation of bile acid, not by excessive deconjugation or decarboxylation of bile acid is not acceptable or desirable. And second one is the bioamine production. What is the bioamine production? Bioamine is sometimes like histamine, tyramine, and cadavine, putrescine, like biological amines which can be produced from the amino acid by decarboxylation during some fermentation process like cheese ripening and the wine production. These bioamines will be produced by the fermentation, by the decarboxylation of amino acid. These bioamines are produced by the probiotic bacteria. So these bioamines are actually very potential carcinogen. So we have to, it can produce cancer. So we have to be careful about that. That this probiotic bacteria should not produce this type of bioamines excessively. So, and third one is the D-lactic acid production. Generally, in our system, L-lactic acid is present, and L-lactic acid is endogenous in our system. It is present in our system, but D-lactic acid is not desirable. Sometimes some lactobacillus can in some at some special situation or condition, they can produce this D-lactic acid, which is the optical isomer of L-lactic acid, and it is detrimental to our body. It can cause the fatigue, nausea, like symptoms like that. So it is not desirable, D-lactic acid production. And the mucus degradation. Now, during the colonization of the probiotics in our intestine or mucus layer, they are sometimes aggressively degrade the mucus layer and damage the mucus layer. So this is also not desirable in this case. And third point is the antibiotic resistance. It is a very important part of the this probiotic pre-assessment of the risk of the probiotics, because generally we know that lactobacillus, lactic acid bacteria, lactobacillus and mainly lactococcus are this may be, one is the antibiotic resistance may be intrinsic, and another is the transferable. What is intrinsic? Now I am explaining it. Intrinsic means this antibiotic resistant property or characteristic of the probiotic bacteria will not be transferred to the other microbiota in our gut or pathogens. Actually, this antibiotic resistance gene are generally chromosomally encoded. In case of lactococcus, lactobacillus, which are considered as a generally recognized as safe by FDA. But some other probiotic bacteria like other genus like Enterococcus faecalis, Enterococcus faecium, and bacillus also, they are they are also the opportunistic pathogens, and they generally, this Enterococcus faecalis, the antibiotic resistance are generally encoded in their plasmid. Plasmid is not the chromosome, plasmid is the extra chromosomal gene in the bacterial cell. A plasmid can be present one or more than 100, and this is the antibiotic resistance gene of this Enterococcus faecalis and Enterococcus faecium are generally encoded in the plasmid, and this is very dangerous because Enterococcus, this, when we are using this Enterococcus faecalis or Enterococcus faecium as probiotic, they can transfer the antibiotic resistance property to the pathogens and the other microbiota in the gut, and then it will be very difficult to control these pathogens by applying antibiotics. When it will become antibiotic resistant, then it will be very difficult. So we have to be careful. When it is plasmid encoded, there will be ample chance of uh, transfer of this plasmid from Enterococcus faecalis or Enterococcus faecium to other pathogens. But when it is chromosomally encoded, in case of lactobacillus and lactococcus, there is no possibility of this gene transfer, antibiotic resistant gene transfer. This is the main thing, and we have to be careful about that. And generally, vancomycin resistant. Vancomycin is considered the last drug applied to kill the pathogenic gram-positive bacteria like Staphylococcus aureus, and these vancomycins are generally encoded in the probiotic bacteria like Lactobacillus and Lactococcus, generally chromosomally encoded. But in case of Enterococcus, this vancomycin resistance gene, this is the ultimate drug. When other drug will be failed, like methicillin resistant Staphylococcus aureus, to kill the methicillin resistant Staphylococcus aureus, and of the some nosocomial diseases which is originated from the hospital. To control these diseases, we have to apply vancomycin drug. But in case of Enterococcus, this is plasmid encoded, and these plasmid are PM 830. In case of Enterococcus, the plasmid, the gene is encoded in this plasmid, and in case of erythromycin, streptomycin, and gentamicin, this drug is this plasmid is PM 831 in case of Enterococcus. So we have to be careful about that. We have to assess the antibiotic resistance of any probiotic before applying it by according to the guidelines of NCCLS. NCCLS means National Committee for Clinical Laboratory Standards. They have given the some standards for the to assess, to judge, to test the antibiotic resistance of this probiotic bacteria, like 12 or 14 antibiotics are generally used, like penicillin, tetracycline, gentamicin, chloramphenicol, vancomycin, like that. And in case of probiotic bacteria, I have told that this vancomycin resistant gene is encoded in the chromosome itself, so there is no chance of transference. But in case of Enterococcus, it is very dangerous. So I hope that you have understood that, and like that, and so this is the intrinsic, means chromosomally encoded gene, and transferable means the plasmid encoded gene. Now the another thing is the we have to be very careful about the some cases of GMO. GMO one means genetically modified organism. Sometimes scientists are applying this probiotic as a vaccine. Some cases, because vaccines are generally attenuated culture of the pathogens which will produce the disease, which are supposed to control that, which are we are trying to control the disease to apply this attenuated culture. This is the normal way of vaccination. But in this case, sometimes there is a chance of happening of this disease again, which will be the bacteria, the attenuated culture is supposed to control the disease. It may happen. So to eliminate this chance, we are generally genetically modified by genetic engineering, recombinant technology. We are inserting the gene in this type of probiotic bacteria uh, to antigen, to produce the antigen and the immune response. So generally this GMO and the products produced from this GMO, originated from the GMO, are not generally accepted by the people, European countries. But still we are using this because the lactococcus, the probiotic, some one type of probiotic, can produce some interleukin 10, which is the anti-inflammatory substances, signaling molecules which can control the inflammation, excessive inflammation. So it is very useful for our say, and this GMO lactococcus can produce this, and scientists are applying. But in this case, one problem is that this lactococcus generally requires this thymine or thymidine for this for their growth, this GMO lactococcus. And in the absence of this thymine or thymidine, they cannot grow and they cannot produce the interleukin 10. This is the one the problem of this. Generally, thymine and thymidine are present at very lower concentration. So this is the one thing. And one thing of this assessment of this risk assessment of the probiotic is this, to properly identify the novel bacteria isolated from any source. This is the first criteria. You have to isolate and identify properly the bacteria or any yeast or any bacteria to be used as probiotic, to assess its risk, the risk associated with it, to assess the risk associated with. Otherwise, you cannot assess the risk properly if you do not identify the bacteria properly. And some identification of this bacteria is done by phenotypic method, means the fermentation of some sugars like glucose, galactose, sucrose, mannitol, sorbitol, raffinose, rhamnose, like that sugar fermentation pattern of the bacteria. You can identify that, but it is very tedious and it is not so reliable. So nowadays the genetic method of identifying this type of bacteria is very popular and very reliable. Generally 16S rRNA primer, gene primer, used for this PCR technique for amplification of this gene and identify the particular bacteria properly, so that we can assess the risk associated with the bacteria properly. So this is the thing, we have to be careful about that. And generally, and now one biomerics company of France has developed API 50 CHL method. It is also the sugar depending on the sugar fermentation pattern, but it is very specific, and they are identify by this method you can identify the, it is also the biochemical test, depending on the biochemical test sugar fermentation pattern of the particular bacteria, but you can identify by this method very easily. So it is nowadays it is used very, this API 50 CHL strips, sugar fermentation strips are used, and this is can be identified by some API lab computer aided program, online program. You can identify the genus and species of this probiotic bacteria. And in molecular technology, molecular technique, by molecular technique, the 16S rRNA is generally genus and species specific. At the species level you can identify the bacteria reliably. So this is the thing. And another thing I want to say from my experience, that generally Streptococcus thermophilus are very good probiotic. We know that. So it is used in the yogurt, and along with the Lactobacillus bulgaricus. But this to maintain the purity of Streptococcus thermophilus is very difficult. This because there are some bacteria like Enterococcus which are may prove to be very opportunistic pathogen. They are almost similar to this Streptococcus thermophilus, and it is very difficult practically to maintain the purity of the Streptococcus thermophilus bacteria, because they are almost same, their morphology, their cell characteristic, all the same, but this Enterococcus faecalis are very robust than the Streptococcus thermophilus. So someday by growing these things, the Enterococcus faecalis will suppress the growth of the Streptococcus thermophilus, and once you cannot get any Streptococcus thermophilus probiotic bacteria, because they are robust, and Streptococcus thermophilus are very fastidious bacteria in this case. So, but they are almost very similar, and they are living with each other very closely. So, very difficult to maintain the purity of the Streptococcus thermophilus, to keep it away, to isolate it from the Enterococcus faecalis. Generally, Enterococcus faecalis are very robust bacteria. They are some antibiotic resistant property like kanamycin resistance, and they are resistant to sodium azide salt. So they are robust than the Streptococcus thermophilus, and they can suppress the growth of Streptococcus thermophilus potential probiotic bacteria. So these are the difficulties generally present, and we have to be careful about that, and so that we can maintain